Alright, so my name is Alexandria Schlosser, and I am going to talk about a recent study conducted on New York City's drinking water. Before we get into the study itself, we are going to follow the path the water would take on its way to the city, from reservoirs and streams through aqueducts and treatments to the pipes beneath the city itself. New York City has a unique water system with a rich history. New York City claims to have some of the best tasting water in the world. Actually, its 9.5 million residents refer to it as the champagne of drinking water. This is because it comes from 125 miles northwest of the city. The Catskill, Delaware watershed is an area stretching across more than 1 million acres. The good taste is said to come from the geology of the Catskill Mountains themselves. The mountains have very little limestone, therefore the water contains low level of bitter-tasting calcium. This watershed provides for approximately 90% of the city's water supply, while the Croton watershed provides for the remaining 10%. This large area of land is protective of the surrounding streams, rivers, and reservoirs. Safeguarding is accomplished through local salt and manure sheds implemented on dairy farms to prevent harmful runoff in the area. Communities near the watersheds also have upgraded septic systems as well as wastewater treatment plants. From the watershed, the water flows through the Catskill Aqueduct. The aqueduct was constructed over a century ago and is considered an engineering marvel. Stretching 92 miles, it takes its deepest plunge at 1,100 feet beneath the Hudson River. Depending on numerous factors, it can take the water as little as 12 weeks to as long as 1 year to make its way from the reservoirs of Catskill to the city. Though this may seem like a long period of time, it is extremely efficient considering this is all done by gravity alone. After water exits the Catskill Aqueduct, it flows into the Kensico Reservoir. It is here that a series of water treatments begin. Robotic buoys float atop the reservoir and transmit information on the water's quality. In this reservoir, fluoride is added to the water for dental health reasons, but more importantly, chlorine is added. Chlorine plays a vital role in the so-called cleaning of drinking water. At a microbial level, chlorine basically goes to war with pathogenic bacteria. When chlorine is added to the water, it forms a weak acid known as hypochlorous acid, which is proficient at killing bacteria such as Salmonella or E. coli. The acid's secret weapon being its neutral charge. As we know, bacterial cell walls have a negative charge and thus repel other negatively charged particles in the water. The acid, being neutral, can breach the pathogenic cell walls and destroy these harmful bacteria. The robotic buoys, as well as field scientists, are constantly monitoring the water's pH, temperature, nutrient levels, and microbial levels. Last year, the buoys recorded 1.9 million measurements, while field scientists collected 15,500 samples from reservoirs, streams, and aqueducts for analysis. From the Kensico Reservoir, the water flows into Westchester County. The Catskill, Delaware Ultraviolet Disinfection Facility is the world's largest, with over 1 billion gallons of water passing through daily. It was in the 1990s that ultraviolet radiation was identified as a safe and effective treatment for drinking water. To be evenly applied, the water must move at a constant speed, allowing for equal exposure. A system of pipes slows the water before it enters one of 56 large containers where UV lights are ready to zap stomach-ravaging microbes. The UV light can kill bacterial cells by damaging their DNA. The light initiates a reaction between two thymine molecules, resulting in an unstable thymine dimer. As the bacterial species undergo longer light exposure, it leads to more thymine dimers. Eventually, too many for the cell to repair. A gene product known as P53 will direct the cell to program death, or as we call it, apoptosis. This treatment is effective in inactivating vegetative and sporous forms of bacteria that are considered harmful in drinking water. Finally, we have the Hillsview Reservoir, the last stop before the city. You can think of this area, the Hillsview Reservoir, as a gigantic bathtub, and by gigantic I mean 900 million gallons worth of water, gigantic. Enough water to supply the city for 1 day. Here, more chlorine is added along with phosphoric acid and sodium hydroxide to kill remaining bacteria, disinfect the water, and raise pH levels, thus preventing corroding pipes and release of harmful metals such as lead. Water tunnels act as drains and whoosh the water down the hill, creating enough pressure to send water to the 6th floor of most buildings. 2 of the tunnels, according to the New York Times Magazine, have been in constant use since they were built, and that was way back in 1917 and 1936. Nonetheless, from here, the water is dispersed through smaller pipes beneath the city. These are referred to as service lines. It is this 6,800-mile-long network of pipes that provide approximately 1,000 water sampling stations throughout the city. Each day, 50 visits are conducted by field scientists as they check the water's temperature, chlorine level, pH levels, as well as other various chemicals. Though the drinking water is tested daily by the city for potential pathogens, the microbial composition has yet to be explored. A team from New York's Department of Health and Mental Hygiene was led by Chief of Environmental Sciences, Inoma Amorigi, to investigate New York City's drinking water microbes and eventually their potential significance to the consumer's health. Though it is well known that drinking water contains a variety of microorganisms, most of which are harmless, the team wanted to characterize the water's microbiome. These microorganisms could have the potential to alter the water's properties, such as taste or smell. For the study, the team took samples from several dozen sites. The sampling sites stretched from the 5 boroughs all the way upstate, thus including both untreated and treated water samples. In the initial analysis, 70 samples were focused on. These samples had been collected from December of 2017 and February, April, and June of 2018. 5 samples of the included were from New York City's cooling towers. To study the water's microbiome, the team used what is called a 16S ribosomal RNA sequencing with Illumina instrumentation. This technology only covers bacteria. Therefore, any eukaryotic organisms, such as amoeba or fungi, were not included. The 16S rRNA gene sequencing has been established as the gold standard for identification and taxonomic classification of bacterial species. The presence of hypervariable regions in the 16S rRNA gene provides a species-specific signature sequence about 5 to 10 bases long. It is found at the 16S rRNA location and acts as an ID tag for that particular bacteria. When the samples are collected, the DNA has been isolated and it undergoes PCR to amplify the 16S ribosomal RNA gene. Then, using a matching system, like we did in lab, the sample's DNA that has been replicated is matched to a bacterium using a genomic library, thus allowing us to know exactly what is in New York City's drinking water. The study reports that in total there were 695 different bacterial species detected. 595 of those species were found in the treated water samples. Yes, a low 100 difference, but many of the species found were reported to be quite common in water systems and or known to be part of environmental processes such as carbon, nitrogen, iron, and sulfur cycle. Some good news for the New York City residents, as the researchers found few enteric bacteria. This includes bacteria such as the notorious E. coli and Salmonella. The team believes that this is due to the safeguarding aspects I mentioned earlier regarding regulations of animal farms and updated sewage systems nearby. However, the analysis did pick up a few types of bacteria that count pathogenic species among them. Legionella is a bacteria species that can be found naturally in freshwater environments like lakes and streams. It becomes a health concern when it grows and spreads in human-made water systems such as cooling towers, which were sampled. Legionella spreads in droplets small enough to inhale. This could in turn cause Legionnaire's disease or Pontiac fever. This is uncommon since an individual would have to aspirate the water, which means the water would have to be accidentally inhaled and travel to the lungs. Mycobacterium was the other bacteria detected. These tiny rod-shaped bacilli require oxygen for growth and have 90 total recognized species. 20 of them are known to cause diseases, including tuberculosis and leprosy. They possess a thick, waxy outer coating that enables them to thrive in aquatic environments. Individuals who have pre-existing conditions such as lung disease, immunosuppression, or HIV are at a higher risk. Infections from these bacteria can cause hemoptysis, lung infection, fever, and other tuberculosis-like symptoms. Non-pathogenic species of both Mycobacteria and Legionella are common in freshwater aquatic systems. The team reports the sequence data was unable to determine whether the bacteria identified in the samples were pathogenic or not. The team included a bacterial community comparison between these three sample types: untreated water, treated water, and water from cooling towers. These percentages show that bacteria species present vary based on location. The treated water and that of cooling towers are related in percentages, while the untreated water is the opposite. This makes sense when you look at the species identified. For example, 70% of bacteria from the untreated samples were identified as actinobacteria. These are common bacteria involved in the degrading of organic matter. Then there is alpha proteobacteria, which are capable of living in low nutrient environments, such as cooling towers. According to the team, the next step is to correlate the bacterial composition with a variety of environmental variables that were measured in the samples. Going forward, it is their intent to expand their research from just bacteria to the analysis of ribosomal RNA from archaea and eukaryotes. Lastly, the study states, in collaboration with ArcBio, the team plans to conduct a shotgun metagenomic sequencing, allowing for functional predictions from the sequences found. Thank you for listening to my presentation. Any information or illustrations presented from this video can be accredited to the following sources.